Understanding CRISPR-Cas9 is easy, actually. What exactly is CRISPR-Cas9? Well, CRISPR-Cas9 stands for Clustered Regularly Interspaced Short Palindromic Repeats and CRISPR-associated protein 9. To really understand the technology behind it, we must examine its purpose. Developed by biochemist Jennifer Doudna and microbiologist Emmanuel Charpentier to enable geneticists and medical researchers to modify parts of a gene by introducing a mutation into the basis sequence of the cell's genome for a certain purpose, whether it be curing genetic errors found in stem cells leading to hematology diseases or agricultural development. Now, how exactly does this work? Cas9, which is an endonuclease enzyme, which you can think more simply of as a pair of molecular scissors, is guided by RNA to CRISPR, which are repeated short clusters found in a genome. From there, DNA is cut. Typically, DNA will attempt to repair the ends using a non-homologous end joining. However, it is prone to genetic errors. However, template DNA or single-stranded nucleic acid is used as a blueprint to instruct homology-directed repair, which has minimal errors, repairing defective genes such as X-linked hereditary diseases. Now, I know all this sounds confusing, but think of it like this. A pothole in the ground, which is CRISPR, acts as a genetic error, which is guided by the driver, the guide RNA, in a truck, the Cas9 protein. To repair the pothole, the truck pours cement in. However, to ensure the amount of cement is correct, a blueprint which acts as template DNA is used. I hope this simplifies it. Peer review is significant to the development of CRISPR-Cas9 to ensure the validity of research by furthering developments and meeting established quality without detracting from the work of other scientists. Peer review significance became evident in the proof of CRISPR's functional viability in human stem cells, enabling it to become what Science Direct depicts it as the breakthrough of the year, by silencing the scrutiny of disbelievers. This is demonstrated as initially the proposal of CRISPR-Cas9's functionality, featuring Charpentier and Doudna, tested the ability to cause point mutations in vitro 9 using Cas9 and tracer RNA and cRNA in the pathogen Streptococcus pyogenes. This was a theoretical proposal regarding its function in eukaryotes and mammalian stem cells. Peer review, however, proved its function in stem cells, highlighted by Feng Zheng's article. In this article, he tests CRISPR on pluripotent stem cells, where he finds introduction of multiple gRNAs can affect multiplex editing of target loci. This demonstrates the ability to use Cas9 in order to change loci, which is the organism's gene structure, ultimately proving the efficacy of CRISPR-Cas9. Now, why is this significant as a form of peer review? Well, it provides basis for the theoretical predictions and evidence, ultimately proving the validity of its technological form. Furthermore, peer review can lead to developments in scientific technology by rectifying flaws. This is highlighted in the discerning of the flaw according to Carl A. Whitney, which demonstrates there is a high frequency of unintended mutations at the edit site, which can lead to phenotypic expressions. The use of peer review enabled the detecting of flaws, which could later be addressed to prevent further complications. Peer review led to the development of technology in this scenario by addressing the unintended cutting of DNA strands, causing unwanted mutations. John G. Gonich and various other scientists addressed this by using sgRNA instead of simply gRNA. Now, what does this do? It prevents the degradation of CRISPR, which increases gene editing accuracy by 90%. This highlights the significance of peer review as it identifies flaws in scientific models, such as the unexpected mutations in CRISPR-Cas9, driving solutions to perfect the technology, ensuring its quality. To conclude, peer review was significant because it enabled the validity of research and the rectification of various errors. The intertwined relationship between science and society is reflected in CRISPR-Cas9's advancement of global health security and the ability to address micronutrient deficiencies in developing third world countries. Exactly is global health security. According to CDC, global health security is the existence of strong and resilient public health systems to prevent, detect, and respond to infectious disease threats. CRISPR-Cas9 in particular aids global health security by characterizing pathogenic agents and being able to assist in the development of vaccines for global outbreaks. A renowned example of this was in the development of the Zika vaccine. CRISPR played an integral role in determining host cell factors, which in essence protect cells from Zika. Once these factors were determined, it allowed the discerning of which genes in the genome should be changed or turned off to protect cells from the Zika virus, ultimately leading to a reduction by 87% globally. This demonstrates the impact of CRISPR-Cas9 on society as it increased global health security by reducing the number of Zika virus cases, allowing definitive aid and assurance should further epidemics or pandemics arise. Furthermore, CRISPR-Cas9 addresses the micronutrient deficiencies in third world countries. Micronutrient deficiencies refer to poor dietary diversities, which have a significant implication, such as anemia, reduced hemoglobin leading to fatigue and dizziness. CRISPR-Cas9 addresses this by altering the gene sequence to increase the nutrient density of foods. This is evidenced in iron enrichment in wheat by the disruption of inositol peniaca 2 incase 1, or IPK1 gene. By increasing the biofortication increases iron density. This is evidence to reduce micronutrient deficiencies in third world countries by 95% since the introduction of CRISPR-Cas9. This highlights that CRISPR-Cas9 was able to address micronutrient deficiency, improving global health. Therefore, the relationship between CRISPR-Cas9 and society is found in its aid to aid global health and security whilst addressing micronutrient deficiency.